professor in the Department of Animal and Veterinary Science. Welcome to our Diverse Stories in STEM speaker series. This is our second seminar in this series. I'd like to acknowledge the student success fee, which made this seminar series possible. And I'd like to thank all of you for being here with us today. Without further ado, I'd like to introduce Dr. Joan Leong, who will be introducing our speaker for today.
Good morning, everyone, and I am very pleased and proud to present our speaker for today, Dr. Adrian Fisher. He was an undergraduate research student in my lab, and he got his first taste of doing research with me back in the day when we were on quarters and there still was a zoology major here at Cal Poly. So let me tell you just a little brief bio. He will be talking more in depth, but he was a student here and he graduated with a Bachelor of Science in Zoology in 2011. And during that time, he took a number of courses, including my entomology course and also a herpetology course with Dr. Chris Lappin. And he shined in those courses. He did so well that on the basis of his demeanor and his seriousness, I asked him to join my lab group. And after he took the course, he said that, yes, he would like to, and that started his journey into doing research. He was a CCRAA apprentice with me, was the Department of Ed, I think, funded program. And so he took advantage of a number of opportunities as a student here. He presented at SCUR and at ARI as well as a student. And so after he finished here, he went on for a PhD at Texas A&M under Dr. Juliana Rangel. And he ended up studying honeybees, which is actually what he started out examining in my lab. But like many students, he had kind of a hard time deciding what area to go into, as many of you might be familiar with. And at one point, he was going to study sun spiders. And so I had a lot of, struggles, but found his way with honeybees. And he got his PhD at Texas A&M, as I mentioned, and then went on for a postdoc at Arizona State University with the lab of John Harrison, and has more recently been promoted to an assistant professor position there at Arizona State, where he teaches, he has his own lab, and he advises undergrad and graduate students, including PhD students. So I am really proud that Adrian has such an amazing record of mentoring students. Since he's left here, he's mentored over 25 undergrad and graduate students in his various capacities. So he has continued the, you might say, wand of helping to make other students' journeys possible as well. And so it was the greatest of pleasure that I could be introducing Adrian, and let's give him a big round of applause.
Can everyone hear me? Okay. So I'm not trying to be shy or anything. I just don't want to get in the way of the screen. So if you don't mind, I'm going to stand in this general area and while I deliver my talk. So thank you very much, Dr. Leong, for that very kind introduction. As you all heard, I did start my journey right here in many ways. This talk will focus predominantly on some of the research I conducted as a postdoc at ASU, but there will be elements of my journey along the way as well. So let me go ahead and get started. And before I launch into some of my research, let me just provide you with a little background about myself. So you saw a little teaser, but these are my parents. So on the left is my mother, way back in the day. And she was born and raised in Mexico before she moved here to the US. My father on the right-hand side was a Vietnam vet serving in the Air Force. And of course, the little chubby baby in both pictures is me. Now, I'm bringing this up because my parents were both very foundational for my background. It was through their guidance that I really can credit being where I am today. My parents were both high school dropouts, but they were also the first voices promoting education and the importance of hard work to get to where you want to be in life. So that really laid the groundwork for me to pursue the various avenues that I ended up pursuing. And that included my interest in animals, particularly the smaller, creepier animals that my parents were not themselves fond of, but they definitely encouraged me to pursue my own interests. And as evidence of this, I'm offering you this crude drawing I made back when I was 8 or 9 years old of a scene that I witnessed in my own backyard that fascinated me, a jumping spider catching a fly. Now, moving on ahead, you heard a little bit about this in Dr. Leong's very kind intro, but to talk a little bit about my college years and some of the experiences beyond, because I'm the first person in my family to pursue a college education and earn a degree, I did have a lot of unanswered questions and uncertainties when I arrived at this stage in my life. And I really did get a lot of clarity right here at Cal Poly Pomona, as you heard, working in the lab of Dr. Joan Leong, where some of my main projects included comparing the foraging behaviors of wild bees, various species of wild bees, including these very neat metallic sweat bees and honeybees, where I generally found differences both in terms of the duration, the amount of time that either major category of bees spent on a flower, but also the revisitation frequency, how frequently they circled back and came back to the same flower. And we interpreted this as having perhaps implications for their effectiveness as foragers. I then continued on by moving on to the lab of Dr. Juliana Rangel at Texas A&M University, where, as you heard earlier, I did have some turmoil in terms of what particular area of focus I would ultimately decide on, but honeybees lured me back in, and I ended up continuing to study these important pollinators, looking at the impacts of various pesticides, including pesticides that, ironically, are applied by beekeepers for the protection of bees, but have these adverse effects on males, which are known as drones, their fertility, and other, an assortment of other effects. I also looked at the impact of various pesticides used in almonds and other major bee-pollinated crops for their effects on worker survival and other aspects of bee biology. After my graduate experiences, I then continued on to Arizona State University, ASU, where I worked with Dr. John Harrison. And here, much of my postdoctoral work was taking a deeper dive into the impacts of fungicides on bee health. This included looking at how particular fungicides impact different aspects of behavior, physiology, and overall health, but also wider colony level effects, which really are a neglected field of study with regard to pesticide impacts on bee health, as I hope to convince you eventually. Thankfully, I've been able to stay at ASU now as faculty, where I continue to pursue questions regarding the impact of different environmental stressors on bee health. But as I mentioned before, I will focus exclusively on some of the work I conducted as a postdoc also at ASU. So you may have noticed the common thread of pollinators being a major point of emphasis in my research through my journey, but let me back up and give you some of the bigger picture details of the importance of pollinators. You're probably aware of some of this, but just to give you more precise details of how pollinators play a role in human health and crop productivity. It's no secret that the human population has seen tremendous increases, particularly in the last few centuries, and quite recently in the last couple of years, the population level has exceeded the 8 billion mark. And this, through that time and to today, has raised a lot of questions regarding food security and sustainability if we're going to be able to long-term feed this increasing population. These questions largely revolve around the use of limited resources, the efficiency by which we use land and water and other important resources, cultivation patterns, and other relevant details regarding our agricultural practices. Now, how do pollinators fit into this picture? Well, pollinators are incredibly important for all terrestrial ecosystems, but in particular, since we're focusing on this question about food sustainability, from the human perspective, we've seen an increasing trend of human dependence on these crops that are highly dependent on animal pollination, including bees and other insects. Now, given this trend of our own increasing dependence on bees and other pollinators, it's striking that, or rather, sorry, let me back up a little bit. I jumped a little ahead. But let me just mention that pollination services, when we combine the activities of bees and other animal pollinators, are all worth annually about $250 to $500 billion. So now to pick up on the point I was trying to make earlier, given the tremendous importance of pollinators to our current agricultural practices, it might be quite striking that we're generally seeing a decrease in pollinator populations and availability across the board, with many species generally declining. So now putting everything together, this trend of increasing, or rather ongoing decreases in pollinator populations are one of the major questions and challenges regarding those issues with crop productivity and overall human food security. So now with this broader background, let's now zoom in on my particular study organism of interest, the honeybee. And I apologize in advance if this slide is a little bit on the simplistic or basic side. I've learned the hard way not to assume that everyone's familiar with basic details about honeybees. So honeybees are a highly social insect, and their society is composed of 3 main castes. We have our 2 reproductive castes, the queen and the drone, the queen being the female reproductive and the drone the male reproductive. Drones, or rather I should say, the term reproductive, as you might have guessed by the structure of the word, refers to those individuals that handle the sole task of reproducing. And drones do this by flying out and attempting to mate with virgin queens from other hives, whereas queens are incredibly important for the ongoing maintenance of their particular colony, as they lay, in some cases, several thousand eggs a day to constantly replace the supply of the other major caste, the workers, which are sterile females that conduct all of the non-reproductive duties for the colony. This includes food collection, defense, tending to the brood and the queen for that matter, and a whole assortment of other behaviors and roles that they perform for their given colony. Now, from the perspective of researchers, workers tend to garner a lot more attention due to this diversity of tasks and interactions that they engage in, both with other members of their own hive, but also in the general environment. We don't really see reproductives going out to gather food. It's more so the workers that are engaged in these more complex ecological interactions and behaviors. So on that note, let me provide you with another key detail regarding the workers. As you might imagine, they are going to be the basis for a lot of the research that I will be discussing. So the typical lifespan for a given worker, and this is particularly relevant to spring and summer. We do see some differences in winter. But a worker, upon emergence, will then spend about the first couple of weeks of her life as what we generally refer to as a nurse. These young workers are domestic. Their job is to clean up the hive. They tend to the brood, and you can actually see a number of larvae, these white C-shaped grubs in many of the cells in this picture. And they're also the ones that will tend to the queen and just engage in other in-hive duties. At some point, these nurses will then transition to foraging behavior. And this is typically the last thing that a honeybee does during her lifetime. This takes her away from the hive, where she then visits various flower or floral resources in the environment, bringing back nectar and pollen, the 2 main food sources for honeybees. And I should mention that foraging is a very energetically costly activity for honeybees. There's a reason why it's typically the last thing they do. They literally work themselves to death, making these continual trips day after day, multiple times a day, and for their body weight, carrying relatively heavy loads back to the hive. And this is important for later, as you might have imagined, why I'm really spending a lot of time harping on this point. But now that we have this basic introduction to the main organism of interest, let's put it in its ecological context, particularly from the perspective of its importance to agriculture. Honeybees are incredibly important for the agricultural practices here in the US and for a number of other countries. Here in the US, their pollination services alone, not factoring in the sales of honey and other hive products are worth a little over $15 billion annually. And in all, they contribute to the pollination of a little over a third of the crops that we use. Now, as I described earlier, pollinators in general are experiencing declines, and honeybees are not immune to this. Although it varies by region, we do see regional declines in honeybee populations, but perhaps the bigger overarching issue are high levels of overwintering colony losses. That is to say that among the managed honeybee colonies in the country, relatively large percentages of them die over winter, and winter tends to be one of the more trying seasons or periods of the year for honeybees, mainly due to in some regions, colder weather and also the lack of resources in the environment. Now, with regard to this issue of decline and the high rates of overwinter colony losses, there are particular environmental factors that are heavily implicated as contributing to these issues in the beekeeping industry. I'm about to show you what are commonly referred to by researchers as the 4 Ps, and you'll see why in a moment. So the first P are parasites. And this is an image of Varroa destructor, the most significant parasite of the honeybee. It can both directly damage honeybees by feeding on their tissues, but also by transmitting a number of viral pathogens. Speaking of pathogens, that is the second P. Pathogens include viruses, different bacterial species, and, as is shown here, different fungal species, all of which can impact honeybee health in different ways and differing levels of severity. The third P is poor nutrition. Now, the image you're seeing here is of an almond orchard. And almond orchards are actually the most highly dependent crop system in terms of needing honeybee pollination. It's estimated that somewhere around 70% of all managed beehives in the country are trucked or transported to almond orchards, which actually are concentrated right here in California, granted in northern and central California, for their pollination, which also quite literally wrapped up maybe about 2 weeks ago. Now, you might be asking, why am I pointing to this as an example of poor nutrition? Well, you may notice that even in this small picture, the only thing you see are almonds. And if you think about it from the human perspective, broccoli is a healthy food item, but if that's all you ate, you wouldn't be a particularly healthy individual. So our current agricultural practices often force managed pollinators like the honeybee to subsist on one type of crop, and thus they do not get their full range of macronutrient requirements. So again, our current practices generally promote this aspect of poor nutrition for managed pollinators. The fourth and final P, and perhaps the most significant in terms of my research, is pesticide exposure. So now let's dive a little bit more closely into pesticides. And as I mentioned earlier, perhaps the biggest issue from the perspective of beekeepers is this ongoing matter of high levels of winter colony losses. This figure is actually a little bit outdated. It's now closer to about 45%, so we are quite literally losing nearly half of all managed bee colonies over winter each year. And while there are a number of factors that can contribute to this, as I described on the previous slide, pesticides are often the most or one of the most cited causes on the part of beekeepers. Now, this term pesticide is an umbrella term encompassing a number of different chemical categories. And these chemical categories are typically designed or at least intended for the control of particular taxa or groups of organisms. Why am I pointing this out? Because not all pesticide categories are regarded equally or have received the same level of attention from a research perspective. For instance, with regard to bee health, the most commonly researched and even from the perspective of the media, the group of pesticides that typically gets or garners the most attention are insecticides, which include the infamous neonicotinoids or neonics. Now, this is not without merit. There have been a number of studies that confirm the negative impacts that various insecticides have on honeybee health. This is by no means an exhaustive list, but it gives you an idea of the wide breadth of effects for individuals, for colonies, and different performance metrics that are impacted by exposure to various insecticides. That being said, as I alluded to earlier, there are other types of pesticides, and they too, to varying degrees, have been implicated in negatively affecting honeybee health. Some of those other categories include herbicides, which are used in the control of weeds or plant pests, fungicides, which, as the name might suggest, are used in the control of fungal pests and pathogens, and acaricides, which are used in the control of arachnid pests, such as mites in particular. Now, for the purposes of my research, I have particularly focused on fungicides. Now, why fungicides? Why this particular group? Well, it turns out that fungicides, at least historically, have enjoyed, if you want to think of it that way, a distinction that other pesticide categories have not. And that is that they were historically considered to be bee safe. That is to say that the mechanism by which they impact the target organisms, fungal pests and pathogens, were not believed to translate well to non-targets, animals in particular, and of course that would include bees. Now, why would any poisonous substance be labeled safe for any particular group of organisms? Well, this rests largely on our standard testing schemes used by the Environmental Protection Agency, the EPA, and other analogous regulatory agencies in other nations, and even here in the US. These testing schemes typically entail taking bees from the field, keeping them in cages in the laboratory condition, and then exposing them to the fungicide or whatever chemical of interest, either by means of spraying them or feeding them known concentrations of these substances. And then from there, the mortality rates, or in other words, how many individuals in each cage die as a result of exposure, is tracked for anywhere from 48 hours to up to 10 days. Now, right off the bat, you might notice some flaws with this approach. There's a very short time period in which effects are monitored, 48 hours typically, sometimes 10 days. Mortality is usually the only metric used, so this doesn't take into account any sublethal effects. If the exposure to the fungicide or whatever chemical makes them sick or impairs them in some way but doesn't outright kill them, that's not typically accounted for in these tests. But also the factor of taking bees and keeping them in laboratory conditions, which neglects the proper ecological interactions and other factors in the environment that could compound or equally affect bee health. That being said, this is our current and past testing regime, and this has had a number of ramifications with regard to the levels of exposure that honeybees face to this particular pesticide group, the fungicides. For instance, a number of studies report that fungicides are the most frequently encountered synthetic chemicals in the environment. And this is both in the context of in the foraging environment, but also as contaminants in the hive of wax, pollen, and other components that bees store or construct in their hive. This itself is largely an impact of a number of fungicides that are approved to this day for application during bloom due to this safety designation. In other words, these fungicides are sprayed on crop systems when they're flowering, when you would expect bees to be present, engaging in their pollination services. Now, to be fair, over the years, there have been a number of studies that have pushed back against this safety of fungicide, or excuse me, this notion of fungicide safety. They have reported negative effects of exposure, either from the fungicides themselves, or more commonly, the ability of fungicides to enhance the toxicity of insecticides and other chemical substances. However, as important as these studies have been to help dispel this notion of fungicide safety, they have generally had a similar flaw to the EPA testing scheme, and that they have almost exclusively, if not exclusively, been conducted in laboratory conditions. So a glaring question regarding a proper understanding of fungicide effects on bee health is the proper field context. How exactly are fungicides affecting honeybee health in field realistic conditions? Before I get into how we went about at least attempting to address this question, let me introduce you to a fungicide formulation known as Pristine, by the trade name Pristine. Pristine has two active ingredients, boscolid and pyroclostrobin, both of which are documented mitochondrial poisons, at least in fungal targets. They impair different intermembrane proteins and ultimately impair or inhibit ATP production. Now, you might be asking, why am I taking the time to introduce this particular fungicide formulation? Well, there are two main reasons. One is that until very recently, literally the last couple of years, Pristine was the most widely used fungicide on almonds and other bee-pollinated crops. So presumably there was a high chance of exposure for honeybees to this particular product. The other main reason is that prior to our own research, a collaborator did take a look at potential effects that Pristine at field realistic concentrations might have on honeybee health, and found that consumption of this fungicide seemingly impaired protein digestion. So in other words, the bees that were exposed to this particular product were nutritionally impaired or deficient in some capacity. So with those two things in mind, let me go ahead and proceed into some of the work I specifically conducted to examine that question posed on the previous slide, looking at field realistic effects of exposure to this particular fungicide. So I'll largely be reporting results from a recent publication where we mainly tested the hypothesis that chronic or long-term consumption of pristine fungicide would negatively impact honeybees at the colony level, particularly in reducing the overall population size, but also altering foraging behaviors, harkening back to that reported protein digestion effect that pristine fungicide apparently had, according to my collaborator. We suspected that honeybees would maybe alter their protein foraging behaviors. And by the way, pollen is the main protein resource for honeybees. So the first step in the process was to devise our treatment groups, and we ended up comparing a non-treated control to 4 concentration variants of pristine fungicide. Now you may notice that 2 of those concentrations are marked with an asterisk, and that is because those 2 represent the lower and upper limits of the field relevant range. So we literally collected pollen, or maybe more so stole pollen from honeybees that were foraging in an almond orchard, and we sent those pollen samples off to be analyzed for residue levels of different chemicals. And the active ingredients of pristine fungicide were detected at such concentrations that we estimated that the entire formulation was present in roughly this range, so between 2.3 to 23 parts per million, or ppm. The other 2 concentrations we used included one concentration that was an order of magnitude below, or in other words, 10 times lower than the lower limit of the field relevant range, and another that was 10 times higher than the upper limit of the field relevant range. Now, we did this because this would have direct implications potentially for the safety of this product. If we saw effects at this very low concentration, this might suggest that even responsible or prescribed application of this fungicide can detrimentally affect honeybee health. Conversely, if we only saw effects at this very high concentration, then this in turn might suggest that responsible or prescribed application of pristine fungicide does not have a significant negative effect on honeybee health, at least in the sense of our measures, and it takes some extreme exposure event to induce such an outcome. At any rate, these were the treatments we ultimately ran with and compared. Now, the other challenge, or there were a couple more actually, was how do we get our honeybee colonies to eat the poison? So we ended up incorporating pristine at those select concentrations shown on the previous slide into pollen patties, which is represented by this trio of cakes here. And these pollen patties are more or less what they sound like, a mixture of ground up pollen, sugar, water, and in the case of the treatment hives, the fungicide diluted in the water and incorporated thoroughly into all of the other ingredients. By the way, we did have our controls that were also maintained in a similar fashion, but of course their patties were free of any fungicides to compare a clean or a safe condition with our various poisoned conditions. Okay, so that took care of one challenge, but the other challenge was because we were imitating field-relevant conditions, our experimental colonies were out in the open. They were able to freely forage and engage in their typical interactions and foraging behaviors and other suite of behaviors. So the other challenge was figuring out how to, as much as possible, limit their access to outside pollen, which presumably was clean, and drive them towards preferentially consuming these treated pollen patties with known concentrations of pristine fungicide. We did so by using a pollen trap system. So this is a model of one of the hives we used. They're typically wooden, but we use these plastic models in part to combat the high levels of heat that they would be exposed to in Arizona, but also because these hives gave us an advantage in terms of trying to filter out that outside pollen from entering the hive. If we pretend to lift this lid and look inside of the box into an empty hive, you would see something like this in the bottom components, where this orange rectangle is highlighting the pollen trap, which was just a panel that sat over the entrance of the hive, and had a number of holes with the diameter precisely measured to allow bees to freely enter and exit. But when they attempted to bring pollen, it more or less prevented them from doing so. And I'll give a more thorough explanation on the next slide. But the end result was that the pollen that the bees attempted to bring into the hive instead accumulated in these removable trays that were attached to the bottom of each hive. So to elaborate on this model a little bit more, let's pretend like we not only take the lid off, but the box itself, so we're only looking at the bottom elements or components of a hive. An incoming pollen forager, note the pollen on her hind leg, will attempt to enter into one of these series of holes, and in the process of doing so, will walk down a short gallery before ultimately emerging through one of the holes in the pollen trap itself. And you may notice already that the pollen has been scraped off. Because bees typically do pack pollen on their hind legs, that adds extra girth. And when they attempt to squeeze in through one of the holes, the perimeter of the hole scrape the pollen off of their hind legs, and it instead ends up in those removable, or rather sliding trays that I described earlier. So this was our way to, as much as possible, restrict them from bringing in outside pollen and drive them towards relying on the patties that we gave them with known levels of pristine fungicide. So hopefully I'm building excitement for the results, but before we get there, just one more bit of introductory material, or rather methodology material. Let me describe to you the experimental conditions in which we conducted this. So this was conducted in the state of Arizona, and while it spanned a number of seasons, much of the work was concentrated during the summer while we wore bee suits, which is a very unique and therapeutic experience in its own way, and I would encourage anyone to partake if you have the opportunity. So now, we're ready to move on to the results, and I'm going to start with perhaps our most striking result using this series of methods, and that is the significant negative effect that pristine had on population levels. It did take a while for this to develop, as you can see through our measure of the adult worker population on the Y over time on the X. But in the latter half of our study, we saw the control group, which in this graph and all subsequent graphs will be represented in blue, diverge significantly, or rather having a significantly greater population level on average than any of our various pristine treated groups and various shades of pink or red. And I want to in particular highlight that one of our groups, which again was that concentration that was an order of magnitude below the field relevant range, also had a similar negative effect in significantly reducing the adult worker population. So as I mentioned before, this would suggest that even quote unquote responsible or prescribed application methods for this particular fungicide was and is detrimental for honeybee health. But it doesn't end there, of course. As I mentioned before, we were also interested in looking at pollen foraging behavior, and we did this by, in two ways. There was our more indirect measure where we gathered up the pollen that accumulated in those sliding trays attached to the bottom of the hive. We were able to weigh that on a weekly basis as an indirect measure of how much pollen each colony was attempting to bring into their respective hive. But the other way was to monitor the entrance of the hive for select times each morning, and we counted all of the foragers coming in, comparing the ratio or the proportion of pollen to non-pollen foragers, and thus compared that measure between our different treatment groups. So I'll show you the results for that, for the second measure I just described, the direct observation of incoming foragers. And here too we saw a significant effect of pristine. Just like with our population measures, it took a while for this to develop, but ultimately we saw significantly greater levels of pollen foragers among our pristine treated colonies relative to the control group. And as a precursor to this study, I mentioned that one of our collaborators discovered that consumption of this fungicide impaired protein digestion by some unknown mechanism. And this result would seem to provide some degree of support for this, and that it appears that these fungicide-treated bees are compensating perhaps for some protein deficiency by trying to bring in more of the pollen, their main protein resource. This result was further bolstered by our second measure, our more indirect measure of pollen foraging, where we compared the amount of pollen that accumulated in the sliding trays between our different groups. And here too, we saw a relationship between pristine exposure and the per capita amount of pollens they tried to bring in, with our various pristine treated colonies bringing in more pollen by population size than our control group, further indicating that perhaps there was some type of protein deficiency in those fungicide-treated bees, and this was a sign that they were trying to compensate for that deficiency. So at this point, we had this trio of prominent negative effects at the colony level for bees exposed to different concentrations of the fungicide. But you may recall from earlier during my introduction that I mentioned that the prevailing or overarching issue for beekeepers is that matter of high levels of overwintering losses. In other words, the high level of colonies that die over winter, which can present an issue not only for the beekeepers, but also in terms of questions regarding pollinator availability for crop production. But does pristine play a role in that at all? Well, it turns out that we were able to compare the overwintering success rates between our control group, and in this case, our pristine treated colonies grouped together or pooled together. And here too, we saw a significant negative effect of pristine, with about 31% of our pooled pristine treated colonies dying over winter compared to 0 losses incurred by the control group. So this might suggest that pristine, and perhaps by implication, other fungicides, particularly those that have similar modes of action, might contribute to that issue of ongoing overwintering mortality or high colony loss rates that has been just such a major issue in the beekeeping industry. So now, at this point, we have this set of negative effects for colonies stemming from field realistic concentrations and, of course, those 2 derivatives of pristine fungicide. But one follow-up question that came to mind was, how exactly is this fungicide affecting worker bees? We did get some insight in that with regard to this question from the work of our collaborator, which again, she found that pristine impaired protein digestion. But we wondered what other impacts this fungicide might have on individual worker health and how these impacts might accumulate to these more pronounced colony level outcomes. In order to get at our question, or rather what we eventually pursued, let me remind you of the typical adult worker lifespan, which again proceeds from newly emerged adult to a 2-week nursing period and eventually to foraging. What we suspected was that maybe for each individual worker exposed to this fungicide, there was some impact on the timing of these key behavioral and life milestone, milestones in life. And we particularly suspected that maybe the bees that were exposed to pristine fungicide had a truncated nursing period and moved on to foraging at a significantly younger age. As I mentioned before, foraging is a very physically taxing activity, and it literally initiates the countdown timer for the end of that bee's life because she'll work herself to death. So if a bee starts foraging at a significantly younger age, this would also suggest that perhaps she'll die at a significantly younger age. And those were indeed our suspicions, that maybe pristine was particularly inducing this effect of what is known as precocious foraging, or foraging at a younger age, and early death in affected honeybees. So we went on and continued, or rather pursued a hypothesis examining this potential impact of pristine. And we tested it by marking with just a dab of paint on the thorax or the back of the bee, several hundred from representative hives or colonies for each of our treatment groups. We were then able to monitor our 2 measures of interest, namely the age of first foraging, by monitoring the entrance of the hive and counting on a daily basis the total number of marked bees we saw clearly engaging in foraging activities. Recall again that the nursing stage is domestic, so we wouldn't expect younger workers to be outside of the hive, and their emergence outside of the hive, and in particular, if they were carrying pollen or some other food product, would be a dead giveaway that they had made that transition to foraging activities. The other measure of interest was any potential effect of pristine on the lifespan, the overall longevity of honeybees. And we monitored this by conducting weekly in-hive inspections, literally opening a hive, pulling out each frame, and counting every marked individual that we saw on each frame in the hive. And we treated the depletion rate of those marked individuals as a proxy for the death rate. And we continued this until we no longer saw any marked bees in any of our experimental colonies. So, using these 2 techniques, let's move on to the results. And the first result, not unlike our colony level effect, we also saw a significant negative impact of pristine on individual workers, as our marked individuals began foraging, or in other words, engaged in precocious foraging at a significantly younger age than the control. And once again, this effect was also apparent in that very low concentration that was below the field relevant range. The controls, on the other hand, were more like that approximate scheme that I showed you on the previous, or a couple of slides ago, where their foraging activities began a little over 2 weeks after emergence, so more in line with what the literature typically suggests for the timing of this event. Okay, so pristine fungicide does induce precocious foraging, but what about survival? Well, it turns out that we also saw a significant effect of pristine on the lifespan and the overall longevity of bees from these various treated colonies. As marked individuals from our various pristine treated hives depleted at a faster rate than marked individuals in the control hives, and ultimately, it may not be as easily distinguishable here at the tail end, but ultimately completely disappeared from their respective hives somewhere along the lines of a week to a week and a half earlier than the last observation of marked individuals in our control group. And this, again, was our proxy for monitoring lifespan, and thus we inferred that the control bees lived longer than the pristine treated bees by a significant margin. So now putting this together, why we suspected that this was such a major issue potentially for long-term colony health is this is our model for our controls, so they fell more in line with the typical scheme for the lifespan of a worker. But our pristine treated bees, as we initially suspected, did have a truncated nurse stage and ultimately moved into foraging and died off at a younger age. Now, the timing of bee development is essential for the supply of workers and replacement generations in a colony, and long-term, these can accumulate to disrupting the colony structure, the availability of the workforce to conduct important duties, and ultimately seems to be an underlying issue for why we saw losses among our pristine treated colonies relative to our controls, which had more of a pattern promoting maintenance and overall growth of their respective colonies. So at this point, we saw that collection of negative effects at the colony level, and now we saw these negative outcomes for individual workers that we interpreted as accumulating to contribute to those negative colony level effects. But we suspected that aside from this aspect of pristine and inducing precocious foraging and early death, that the fungicide may have also induced sublethal impacts on honeybee health that can further contribute to why we see high losses and reduced population levels. And we suspected more specifically that maybe pristine was affecting honeybee cognitive abilities. Now, I'm not sure what everyone's background here is with regard to honeybees or insects in general. Perhaps you don't necessarily think or believe that honeybees or insects can learn and retain memories. But if you're of that mindset, let me just run through a brief exercise to persuade you otherwise. So let's take this scenario where we have a honeybee hive, and we have an individual from this hive, a forager, that discovers a novel food source, so this flower patch somewhere in the environment. She'll spend time visiting the various flowers here, maybe collecting some of the food resources, and eventually make her way back to her hive. Now, what happens in the hive that supports the idea that honeybees are indeed able to learn and remember key aspects of their environment? Well, if we took a peek inside of the hive, we might see something like this, a behavior known as the waggle dance, which is characterized by this looping figure-8 motion and a gyration in the center of the dance. Let's, if we can, take a look at that again. So we can imagine that the dancing individual is that forager in the scenario, and again, she's engaging in this looping figure-8 motion and a gyration in the center of the dance. Now, as you might have guessed by looking at the nature of that dance itself, it's not exactly a random motion. But what does it tell us about cognitive abilities in honeybees? Well, it turns out that there are certain key bits of information encoded in that dance itself. For instance, that dancing bee is able to communicate to her nestmates, to her sisters, the direction of the food source, which they are able to point out relative to the position of the sun in the sky. Encoded in that dance is also the distance or the approximate energy expenditure to the food source. And last but not least, she can communicate how excited she is, how good or she deems that particular food source to be to her nestmates. And that can affect the number of her nestmates that are recruited to also go and investigate and potentially collect from that particular flower patch. So putting this all together, I hope you are now convinced that honeybees are able to learn, and they demonstrate this by their assessment of their flower patch, investigating how good it is, and then ultimately taking back maybe samples of the food these flower patches have to offer to their hive. And then, of course, upon return, their ability to communicate their recollection of their assessment of that flower patch to their nestmates supports the idea that honeybees are able to retain memory and, more importantly, communicate those memories to their nestmates to also get them to visit those food resources. So this led us to our second follow-up question, considering if our fungicide of interest, pristine, does in any way impair the cognitive abilities, the learning and memory capabilities of affected honeybees. So I will now report to you the findings of another recent publication, in this case led by a recent graduate at ASU, where we tested the hypothesis that, more particularly, that pristine fungicide consumption would negatively impact the learning and memory capabilities of honeybees. So, as I mentioned before, hopefully if you were of the mindset that insects cannot learn, you are now convinced by my quick and dirty example, but you might be asking, how exactly do you test learning and memory in honeybees? In order to get at that, let's revisit the work of Ivan Pavlov. Now, Pavlov is, of course, famous for his work on dogs, where he devised this conditioning test composed of three main phases, including the preconditioning phase, where dog subjects were exposed to two different stimuli, one of which was biologically irrelevant, a ringing bell, and the other of which was very relevant and typically induced a visible, marked response from the dogs in the form of salivation and general excitement. This preconditioning phase of the test was then followed up by the conditioning phase, where the dog subjects were co-exposed to both stimuli, typically in the order of the previously irrelevant stimulus, the ringing bell first, followed shortly afterwards by the food. And the idea here was to test the ability of dogs to learn that a ringing bell would lead to food, or the association, rather, between what was previously irrelevant to them with what is definitely very relevant for them. Pavlov then followed this up with the post-conditioning phase, where he could test the memory, the retention of this learned information on the part of the dogs by exposing them to the ringing bell and observing if they indeed responded to this ringing bell alone without food, suggesting that they remembered, when I hear a bell ring, that means I'm going to get food. So they got excited just in response to the ringing bell. We can use a similar overall scheme for honeybees, in this case termed the proboscis extension response, or PER test, to also assess learning and memory, but it has many of the same components. For instance, in the preconditioning phase, honeybees, when restrained and harnessed in the lab, are then exposed to two different stimuli, one of which is more or less irrelevant to them, or at least doesn't draw a clear response, some odor that is puffed or wafted past their antennae. The other of which is very relevant to them, a droplet of sugar water, for instance, and elicits this very typical response where they extend this tongue-like mouth part known as the proboscis. We can then proceed with the conditioning phase of this testing scheme, where we co-expose them to the two stimuli, not unlike the dogs being co-exposed to those two stimuli. And this also typically proceeds with first the puffing of that odor past the antennae, followed up by the administration of a droplet of sugar water. And similarly, we're testing to see if they're able to learn the association between smelling the odor and receiving a food reward. Sometime afterward, not unlike Pavlov's original test, we can then conduct the post-conditioning phase, where we now only expose these test subject bees to the odor to see if they respond similarly to when they were receiving food rewards, indicating that they learned that when I smell that odor, I got a droplet of sugar water, so I'm going to anticipate this by extending my mouth parts, expecting to be fed. Now, I won't leave you with just this more abstract description. Let's look at a quick video to see how this works. So note the lights at the bottom. When the light switches to the far right, the odor is on, and that bee is then immediately given a droplet of sugar water. So this is the conditioning phase of the experiment. And now we can proceed into the post-conditioning phase, where now it's just the odor. And when the light switches, that individual responds by extending her proboscis, indicating that this particular individual did learn that association and is expecting to be fed even though she's only exposed to the odor. So now, using this testing scheme, we were able to assess bees from our different treatment groups to compare and contrast between potential impacts of the fungicide on learning and memory capabilities versus the control. And keeping up with the theme that we've seen so far, here too, we also saw a significant negative effect of pristine fungicide. In this case, in particular, among the two higher treatment groups, but this nevertheless entailed the upper end of the field-relevant range. So even at field-relevant concentrations, albeit to a limited extent based off of our findings, this pronounced negative effect of reduced cognitive abilities could also be induced relative to bees that are not exposed, or at least exposed to lower concentrations of the fungicide. So let's put everything together now, and it paints somewhat of a grim picture, starting first with our interest in pristine fungicide on the basis of its common usage, at least until a couple of years ago, and some implication for how it affects bee health based on the work of our collaborator, we proceeded with exposing honeybees to this fungicide at field-relevant concentrations in field conditions and saw a number of cascading negative effects for bee health. Starting first in somewhat reverse order with our impacts on individual workers, we again saw precocious foraging or foraging at a significantly younger age among bees that were exposed to different concentrations of this fungicide, and also the reduction in cognitive abilities, learning and memory capabilities in bees, particularly exposed to some of the higher concentrations, but still nevertheless encompassing to some extent the field-relevant range. These two factors, or at the very least precocious foraging, seem to be the underlying factor for the reduction in lifespan, which we also saw in honeybees that were exposed to different concentrations of pristine fungicide. And ultimately, this accumulation of negative impacts for individual workers seems to be the underlying factor for why we see reduced population levels over time when exposed to this particular fungicide and perhaps by implication other fungicides, especially those with similar modes of action. Now, not shown here are our overwintering results, but you may recall also that we saw a significant reduction in overwintering success among colonies that were exposed to pristine fungicide. And so this would also fit in this general cascade of negativity associated with exposure to the fungicide. So at this point, it may seem like a very bleak, grim picture, but I do want to offer maybe some good news or a silver lining, some of the good that's come out of these discoveries and those made by our colleagues and other researchers. And that is increasing discussion and dialogue centering around better, or rather improvements in how we assess and approve pesticides, and ultimately for the enhanced protection of pollinators and human food security. And I've been fortunate to be a part of some of these efforts. These are a couple of publications in which I worked with a diverse group of researchers representing 6 different countries, and we were able to put our heads together and notice the commonalities. And I should mention that not all of the researchers on these teams necessarily focused on honeybees or the same exact chemicals, but we were struck by just the reoccurring theme of negative effects on various pollinator species conferred by different approved and regulated chemical products. And so we were able to combine our efforts and not only address some of the shortcomings in current regulatory policies, but also in some cases suggest solutions and improvements that would go a long way towards these goals of protecting pollinators and improving food or crop production. All right, so let me kind of bring us back to the beginning. You may recall from the title of my talk that I more or less promised some kind of correspondence between myself and my study organism, or some parallel between my own journey and what I see with these discoveries and studies in honeybees. But how exactly does that work out? Well, at the very beginning, upon emergence, we have certain expectations and limitations, but also opportunities and different chances to excel in ways that we may not have expected at the start. For honeybees, some of those limitations include the 4 Ps, as I mentioned earlier, different environmental stressors and other factors that can limit their health and growth and effectiveness in their pollination services. But I'd like to think that my colleagues and I, have played our small part in helping to push along the solutions in suggesting different ways that we can improve circumstances for honeybees and other pollinators by reforming the way we test and approve pesticides, and just general suggestions for enhanced protection of pollinators and, by extension, human food sustainability. Perhaps particularly for the students, but really everyone here could relate to this, but maybe along the way we all face our own toxic elements in our environment, whether it's external factors that can drag us down or discourage us from our pathway, but also some internal doubts and uncertainties that can also distract from what we ultimately aspire to do. And in this regard, and I hope that all of you can say the same, I've had a lot of great mentors that have helped to combat some of these toxic elements in the environment and helping to open doors and enlighten me on different opportunities and paths that I could take to overcome various obstacles. So now when I consider where I'm at now, thankfully faculty at ASU, and what I ultimately want to accomplish long-term in my current position, I find that I more or less have the same outcome or goals as my study organism, the honeybee. Just as a honeybee collects food resources for her colony, I find that it's also all about the colony. It's about your particular communities and the positive impacts, even if it's just in a small way, you can have with those you interact with. And I hope that, particularly for the students, as you're continuing through your journey, you're able to identify your colonies and the communities you can impact, whether it's those you are born into and those you eventually become a member of through shared interests and just overlapping concerns as you discover your own pathway. So I don't know how I'm doing on time, but this is more or less what I have for you, so thank you very much. And yes, this is a student that was attracted to a swarm for some reason. I'm not entirely sure why. Yeah. I think there's a mic coming around.
I'll go far away from it. Yeah, for your different treatments for the like pesticide, how did you account for like bees and control like which patty that they were choosing? Was it like within the colony, or like how did you account and control which patty that they were selecting for which colony that they were a part of?
Yeah, that's a great question, and it's not something that I mentioned, but these patties were put inside of the hive, because if we put them in the environment and they collected from them and attempted to bring them into the hive, they would face the same issue with the pollen trap. So these were placed in gaps inside of the hive so that young workers could access them and consume the pollen directly.
Okay, I don't know if this question makes sense for this, but I was wondering if seasonality is like an influencer in the study, because I know you mentioned like foraging season like has its own time, but it seems like it was at least like a year study, because I saw like there was one figure that was like February to like the summer, I think, and then another one that was like May to November. So I was wondering, did you have like a different question for like each different season, or like in terms of like, I guess, honeybee or like worker population, like measuring that, was it like over the entire year, or like just like over a few seasons?
Yeah, that's a great question. So a follow-up study that we conducted that I didn't show here was trying to take into account any impact of season, because there are significant differences in the physiology and behavior of the bees themselves in winter, for instance, versus summer or spring. They tend not to eat as much pollen, so we would expect if we gave them the same treatments that they would eat a lot less of it and maybe have a reduced impact of exposure. Also, in general, we try to mimic the timing for exposure to in almond orchards, which was our reference for the concentrations we use. So we try to skew our study to start more in spring, because that's about when we would see the beginning of any impact of fungicide exposure. But that is a great question. There definitely could be a seasonal component, and we did, to some extent, take that into account in a later follow-up study.
Have you had any reactions from the fungicide manufacturers?
Yeah, that's a great question. Thankfully, nothing hostile. We've had a few requests to meet or interview by BASF, which is the main producer for Pristine fungicide. Although internally among my colleagues, we kind of joked that the reason why Pristine was more or less pulled from the market was our work, and maybe they have a dartboard with our pictures on it that they're kind of cursing at us, but nothing hostile. It's just been a few individuals that have reached out to us just for more information and to talk with us.
Did you treat for or notice any parasites, mites, diseases inside the hive? And did Pristine have any effect on those?
Yeah, that's also a good question. So typically, mites are assessed and treated for at select times of year, usually in early spring and then in fall. And our study fell between those time periods. So there could have been residues, perhaps, of what we used in early spring. but since all of the treatment groups would have received those treatments, we assumed that maybe wasn't a factor that compounded the impact of the fungicide. We didn't tightly monitor the population of mites, but in part because we have those particular time points where we check their levels and at that time act if needed.
Oh, hello. I really liked your presentation. I did want to ask, though, for one of the studies mentioned, especially the one talking about like the learning ability and the memorization ability of bees, for the 0.23 parts per million results, they were very similar to the control group, which isn't usually seen in like the other research shown. Like there was a very substantial difference in terms of like the effectiveness between the control group and just like any amount of fungicide. So I was just wondering, why is it that in that specific study, the small amount of fungicide seemed to align pretty closely with the control group and at times even sort of surpass the control group, especially in learning? I was just like, that's kind of, yeah.
Yeah, that's a very good and interesting question as well. So we still don't have a clear understanding of the transmission of residues of the fungicide within the tissues of the bees themselves. We suspect that because they are consuming these tainted pollen sources, that there's maybe a more direct impact on the gut, and that's what induces the impairment of protein digestion, and maybe by extension, some of the resulting behaviors and altering pollen foraging behavior and maybe precocious foraging. But we don't know what, if any, residues make it to the brain, and that would maybe exert a more direct impact on the learning and memory capabilities. So perhaps at the higher concentrations, there's enough of it that it does ultimately, to some degree, transmit to the brain, but maybe not so much with the lower concentrations, which is maybe why we were not seeing differences between those lower concentrations and the control versus the differences in behavior and lifespan, which probably stem from that more direct impact of the fungicide on gut tissues and maybe the surrounding organs.
Thank you for coming today. I just wanted to ask if you had, in contrast to having darts thrown at you by the fungicide industry, are you being celebrated or encouraged by the almond corporations?
Right. So I would say, just like Chris's question, it's been somewhat neutral. I've had some individuals from the Almond Board of California, for instance, that also have reached out for more information, but not necessarily promoting our research or taking anything away from it either. So it's been kind of quiet on their end, but mostly neutral, just those initial contacts.
Hi, thanks. Did you look at exposure of pristine to the drones or the queens in the colonies at all? And do you suspect if they had any impact on them?
Yeah, that's a good question. We didn't look at the effects on the reproductives. I didn't really mention this necessarily, but the pollen patties were exclusively consumed by young adult workers. They need protein resources to build up their flight muscles, and then they in turn will feed the queen and larvae with a jelly they make from the protein resources they consume. So they're in a way a filter for the young and the queen. That being said, there may have been some degree of transmission that we didn't account for. Some results I didn't share were the population levels of the brood, and we ultimately didn't see any differences in the egg laying rates between the queens. So at least in that major function, there was no clear indication that it impacted queens. Drones are transient. Their presence in the hive is dependent on other factors, and some hives did not produce any drones at all. So since it was not a consistent element in our study, we also didn't take into account potential effects on that demographic.
With the development of things like biopesticides and overall just utilizing fungi and different things that are naturally occurring in the soil for these same treatments, how do you think that could change overall population and production?
Right. So, well, depending on the particular species or products, you could imagine them being an improvement or perhaps inducing similar negative outcomes. I think part of the drawback is, to this day, these synthetic chemicals are the most effective, at least in the moment, but as we're seeing, they can have long-term ramifications on non-targets, and then we have the issue of resistance on the part of the target organism. So it really is a complicated web, but you could imagine that more concentration on developing those alternatives could be a big benefit in addressing some of these concerns with pollinator health.
You mentioned that you worked in collaboration with 6 other countries in a few of your other research projects, and I assume that, or you mentioned that they were all different pesticides and different pollinators for each one. Did they all similarly align with how pristine affects honeybees, or did they use like different methods and capture different results?
Yeah, so mainly different methods. I don't know how well I emphasize this, but there really isn't a big emphasis on looking at field relevant effects. Some researchers still use the general model of testing the effects of various chemicals on pollinators in laboratory conditions, albeit not necessarily sticking strictly to the standards by the EPA or whatever analogous regulatory agency in different nations. So we were a little bit unique in the fact that we really had that strong field component, but even in their assessments, using different pollinator species and chemicals, and also in some cases focusing on different metrics of the impact, they also generally report negative effects on their species of interest.
One more question? Who wants to ask our concluding question? Yes. Last but not least.
How big a problem are their fungal diseases?
It varies. One particular species, and actually it was the picture I showed you on here, it's known as Nosema serrani. It's probably the worst. It's not quite on the level of Varroa in terms of its impact on honeybee health, but it seems as if
I meant on the crops.
Oh, on
like
the
if you're going to
crops.
reduce using these fungicides, is it going to have a huge impact on crop production and the businesses?
Oh, yeah, that's a great question. So it does vary by environmental conditions. So these fungicides tend to thrive when it's a particularly wet year. Actually, I literally saw this a couple of years ago, but in drier conditions, some growers avoid spraying altogether, and that would be beneficial, but you can't necessarily rely on it being dry and to the point where fungal pathogens are not as much of a concern. So I don't know if I can provide you with an exact figure, but in wetter conditions, they can be a far bigger nuisance and a detriment to crop productivity.
Hi, everyone. So before I go ahead, I just want to give a round of applause to our speaker. I hope that you all took a lot out of this event. This is my first time attending, so I just want to say that I was really excited to see firsthand the learning opportunities that are provided to you all as students. I work here at the Annual Fund. I'm one of 2 associate directors, and part of my job is raising funds to help support you all and your educational journey here at Cal Poly Pomona. So I want to thank the team at the College of Agriculture for putting this on, and Bill for inviting me today. The Diverse Stories in STEM program participated in our campaign last year to help raise funds to help put on events like this for you all. And last year they ended up raising about $400 with the help of 2 staff members and 1 student pushing the campaign. Well, I think that's great, right? That's $400 that they didn't have before. I definitely think that there's an opportunity to make a bigger impact, right? So like our presenter said, it's about the colony, right? And the impact of the colony. I want to provide you with the opportunity to help as well, to help provide more opportunities like this for students to continue to benefit, to learn, and hopefully to inspire to make change in our world. So you all can help. The Diverse Stories in STEM is participating in our Giving Day campaign this year. And they are hoping to raise more funds to provide more events like this for you all. Quick show of hands, has anybody ever heard of Giving Day or participated before? Okay, we got a couple hands, nice. Yeah, so Giving Day is a great opportunity to first and foremost showcase the impact that Cal Poly Pomona is making, right? We wanna first and foremost celebrate all of the exciting things that are happening here at Cal Poly Pomona. And we need your help to show that to the world, show that to your friends, your family, your personal network. So that's the way that you all can make an impact by sharing your experience, sharing how diverse stories in STEM has impacted you, how you have benefited from it, and encouraging your folks to support, right? So you all can sign up as an ambassador. I have a couple of flyers here that you all can take with you. All you have to do is just scan the QR code and with your CPP email sign up, and we have videos that you can see to help you get started. We have tons of resources. We have a lot of sample messaging templates. So we don't expect you all to be experts at this type of thing, but we have a lot of tools to help you be successful and to help diverse stories in STEM get the funds that they need to continue to do opportunities like this. So feel free to grab a flyer. I'll be posted up in the back after the event. If you'd like to connect more, feel free to reach out to me as well. Yes, Bill? Yeah, good question. So my name is Jorge Garduño. Again, I'm one of 2 associate directors at the annual fund, and Giving Day is happening on April 17th and 18th. So during those 2 days, we have 36 hours to raise as much funds as we can for different parts of the campus, and with your help, hopefully we can help generate more funds for diverse stories in STEM. Thank you.
Thank you all for attending our second Diverse Stories in STEM. I want to give Dr. Adrian Fisher another round of applause. Thank you so much. And again, thank you so much to the SPICE grant, as well as my collaborators. I'm Janelle Ortiz, Rachel Blakey over here, Imana Taras. Where did you disappear to? There you go in the back, helping build this program. And hopefully we will see you all again next semester for our next presentation. Please stick around. There's food in the back for you all. And thank you all. Have a great rest of your week.
I appreciate it.
That was so